In the last class, we discussed about different parts of the bacteria, and we already discussed about that outermost part of the bacteria, that is the glycocalyx layer. After that, we have discussed about that cell wall structure and the composition of the cell wall. In different types of the bacteria, and different compositions are present. So we have discussed about all the types of the components, compositions of the bacterial cell wall. Coming to the next topic is classification of bacteria based on chemical composition. Classification of bacteria based on chemical composition of cell wall. Total here, bacteria can be classified into two types. Based on cell wall composition, here bacteria are classified into the two types. There is a gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria and gram-negative bacteria. See, this classification especially done by the gram staining technique. The gram-positive and gram-negative bacteria classification done by the gram staining technique. Before discuss about the gram staining process, we have to discuss about what are the chemical composition present in gram-positive bacteria, or what are the chemical components are present in gram-positive bacteria, and what are the chemical components are present in gram-negative bacteria. We discuss first. So, in that case, gram-positive bacteria. Within gram-positive bacteria, there is a thick cell wall is present. The cell wall is a single layered structure. Totally, this is the cell wall. The cell wall is single layered structure. The cell wall is single layer structure. And coming to the gram-negative bacteria, within this case, the cell wall is thin and it forms in two layers. Especially the gram-negative bacteria, the cell wall, it's having two layers, not single layer. It's having two layers. But in gram-positive bacteria, only one layer. So, the thickness of gram-positive bacteria more than gram-negative bacteria. So, this is the gram-positive bacteria cell wall having more thickness, and this is the gram-negative bacteria. The cell wall thickness is less compared with the gram-positive. And see, this is a single layer structure, and the cell wall here in gram-negative bacteria forms in two layers. Here only single layer, here two layers are present, right? That is the difference between, that is the first difference between gram-positive and gram-negative bacteria. And I already said the thickness more in gram-positive, less in gram-negative. The thickness is here 20 to 25 nanometers. 20 to 25 nanometers thickness seen in gram-positive bacteria. In gram-negative bacteria, 8 to 12 nanometers. In gram-negative bacteria, 8 to 12 nanometers. So, more thickness in gram-positive bacteria, less thickness in gram-negative bacteria. And mucopolysaccharides. Remember, here polysaccharides, that is indicates carbohydrates. Mucopolysaccharides, these are modified carbohydrates or modified polysaccharides. Which are present in gram-positive bacteria, the mucopolysaccharide percentage is 70 to 80%. In gram-positive bacteria, mucopolysaccharides percentage is 70 to 80%. And gram-negative bacteria, the percentage in gram-negative bacteria, the mucopolysaccharides percentage is 10 to 12%. Here 70 to 80%. Here 10 to 12%. So more percentage of the mucopolysaccharides are present in gram-positive bacteria, less percentage of mucopolysaccharides are present in gram-negative bacteria. Teichoic acids. These are very special structural components which are only seen in gram-positive bacteria. So teichoic acids are present in gram-positive bacteria. Teichoic acids are absent in gram-negative bacteria. So this is the structural differentiation between gram-positive and gram-negative bacteria. See here, I like to discuss about the very important point is here only single layer. Here cell wall forms in two layers. Here only single layer structure. Here two layers are present, and thickness is more. Here thickness is less. Right? So that is the differentiation between gram-positive and gram-negative bacteria chemical composition. Then we will discuss about how we are identifying the gram-positive and the gram-negative bacteria. After differentiation of gram-positive and gram-negative bacteria, we'll discuss about the gram staining process. Before going to discuss about gram staining process, what is the use of gram staining process? See, generally we are unable to identify the bacteria by the naked vision. We are unable to identify by the bacteria by the naked vision. See, what I am telling here, we are unable to identify the bacteria by the naked vision. How can we identify the bacteria cell wall? You are unable to identify. Then, in that case, one of the scientists introduced one technique. The technique is called as gram staining technique. That gram staining technique is very useful for the differentiation of gram-positive and gram-negative bacteria. In that case, the scientist's name is Christian Gram. Christian Gram. That's the reason why that technique is called as gram staining technique. In that gram staining technique, see the gram that is the gram that is completely related to the scientist name. See, staining technique. We know stains. Stains are nothing but coloring agents. Stains are nothing but here coloring agents. So in this process, we are using different types of stains. That stains are supporting for the identification of gram-positive and gram-negative bacteria. See, very first step, we have taken one of the glass slide. For suppose this is one of the glass slide. We have taken glass slide, and the surface of the glass slide we have taken bacterial culture or bacterial smear. We have spread it. We are spread it. That bacterial culture spreaded by the help of loop. In the laboratory, in microbiology laboratory, loops are there. By the help of loops, we are spreading the smear, bacterial smear. After that spreading process, the bacteria, the slide exposed to the heat. For suppose this is one of the burning candle or burning one. So that glass slide is exposed to that burning candle. Then whatever that bacteria cells are present surface of the glass slide, by the heat fixation, the bacteria cells are attached to the glass slide. So that process is called as heat fixation. Very simple. Heat fixation is nothing but inside, there is a laminar airflow, one of the equipment. Inside that laminar airflow, there is a burning candle is there, or Bunsen burner is there. So we have taken that bacterial culture on the surface of the glass slide, we have spreaded it. If the slide exposed to the heat, due to that heat, the bacteria cell walls attached to the slide. So that step is called as heat fixation. After that heat fixation process, we are adding one of the coloring agent. See the very first time, that is the crystal violet. Crystal violet is the first coloring agent in this process. Crystal violet, the color is purple color. Crystal violet color is the purple color. So crystal violet we are added. Once we are added that crystal violet reagent or crystal violet stain, then the cells are modified into purple color. See, we don't know because we are not identified that cells under the microscope. We are added the crystal violet. After that, next step is we are added the grams iodine. After the addition of crystal violet, we washed out that slide very gently. After that washing process, next one we are added the grams iodine. What is the use of grams iodine? For suppose this is the bacteria. After the addition of crystal violet molecules or crystal violet reagent, this is one of the crystal violet molecule. It's having purple color. So the crystal violet molecules are binded to the bacteria cell wall, especially the crystal violet molecules are binded to the cell walls of the bacteria. After that addition of crystal violet, we added the grams iodine. For suppose this is one of the grams iodine molecule. After this addition of crystal violet, if we add that grams iodine, it increases the affinity between crystal violet and the cell wall. For suppose this is the cell wall and this is the crystal violet molecule. So between these two, the affinity, the binding affinity enhanced by the grams iodine. So what I am telling here, this is the bacterial cell wall and this is the crystal violet molecule. So after addition of crystal violet molecule, if we add the grams iodine enhances the affinity between cell wall and the crystal violet molecule. So crystal violet molecules are very strongly binded. Crystal violet molecules are very strongly binded. So after that, addition of grams iodine. Next one, we are adding 70% of the alcohol. We are adding 70% of the alcohol. Then here alcohol is the decolorizing agent. Here alcohol is the decolorizing agent. Why this one is decolorizing agent means generally, okay, this is the cell wall. This is the cell wall. This is thick cell wall. If we add crystal violet molecule, these are the crystal violet molecule. So if cell wall is thick, okay, that one, the cell wall unable to destroyed by the alcohol. Thick cell walls are not destroyed by the alcohol, right? For suppose this is the thin cell wall, and these are the crystal violet molecules. These are thin cell wall. The thin cell walls are destroyed by alcohol. The thin cell walls are cell wall layer removed by the alcohol, because the thin cell walls are easily diluted within the alcohol. But this one is the thick one. This is unable to disturb or unable to distract by the alcohol, because this one is the thick layer, this one is the thin layer. So here whatever that thick cell wall contains structures are there, these are not distracted by the alcohol. But whatever that thin cell walls are there, these are easily distracted by the alcohol. After that, after decolorizing agent, then we are adding the second stain. The second stain is the safranin. First stain is here crystal violet. The color is purple color. Here second stain is safranin. Safranin, the color is red color. Saffron color is the. After the addition of safranin, the cells will get another color. Mostly we don't know. After that addition of safranin, we will wash out that slide. Okay, gently. After that washing process, if we observe the slide under the microscope, the bacteria cell, some cells may be exhibit one type of color, and a group of cells are exhibit another type of color. See, if the bacteria cells are exhibit purple color, that group of bacteria are called as gram-positive bacteria. If the bacteria cells are exhibit red color or pink color, okay, these bacteria are called as gram-negative bacteria. So here after that addition of safranin after the washing process, right? If the slide observed under the microscope, if the bacteria cells exhibit purple color, that bacteria are called as gram-positive bacteria. If the bacteria exhibit red or pink color, that bacteria are called as gram-negative bacteria. So during this process, how the color variations takes place. We already discussed about there is a gram-positive bacteria. Having thick cell wall. Gram-positive bacteria having thick cell wall. What I said here. First of all, we added crystal violet. If the cell wall is thick, that is not distracted by the alcohol. If the cell wall not distracted by the alcohol, so already one of the stain is binded to the cell wall. That stain is crystal violet. So here gram-positive bacteria exhibit purple color. Remember, gram-positive bacteria having thick cell wall. Thick cell walls are not distracted by the alcohol. Already thick cell walls are binded with crystal violet. Crystal violet color is purple color. Gram-positive bacteria color is purple color. So that is a simple explanation. Coming to the gram-negative bacteria, within that gram-negative bacteria, there is two layers are there. This is the first layer, and this is the second layer. First layer binded with the crystal violet molecules. After the addition of crystal violet molecules, after the addition of decolorizing agent, see, the outermost layer will be washed out. So here there is no crystal violet molecules, because outermost layer, okay, removed by the alcohol. Then after that decolorizing agent, we are adding the safranin. We are adding the safranin here. We are adding the safranin here. So here this is one of the, okay, these are the safranin molecules. These are the safranin molecules. So safranin molecules are binded here because outermost layer is washed out. Along with the outermost layer, the crystalloid molecules also washed out. So freshly here, safranin molecules are binded. The safranin molecules are binded to the second layer. Then the gram-negative bacteria exhibit red color or pink color. Red color or pink color. So this is the gram staining process. By using this technique, we are separating the gram-positive and gram-negative bacteria. This technique first introduced by the Christian gram scientists. Remember, here we are using two types of the stains. The very first type is called as crystal violet stain. That stain exhibit purple color. The second stain is the safranin, right? The safranin exhibit red color. So by the usage of these stains, and there is a gram iodine is there, and the decolorizing agents we are using. So we are performing the gram staining process. By the gram staining process, we are separating, or we are differentiating the gram positive and gram negative bacteria. So that is about the cell wall. That is about the cell wall.